In this video, we will look at a critical step of a PCR that you need to carry out before you have entered the lab, and that is PCR primer design. The design of the primers used in a PCR is critical because if you get this step wrong, then at the end of a PCR, the product in the tube will not be the DNA you need. And this means you've wasted time and reagents. Typically, when designing a primer, you take into account three things. 1. The melting temperature of the primer. 2. The possible formation of structures such as hairpin loops and primer-dimer pairs that could disrupt the reaction. And 3. The specificity of the primers. Before we go any further, we need to discuss two types of primer temperature: the annealing temperature and the melting. The annealing temperature is the middle temperature of one cycle of the reaction at which the primer sticks to the denatured template. The melting temperature, or TM, of a primer is the temperature at which 50% of the primer would melt to form single-stranded molecules if it were double-stranded. The relationship between the annealing and melting temperatures is that you usually set the annealing temperature on the PCR machine to 3 to 5 degrees centigrade below the melting temperature. The TM of a primer is determined by the A's, T's, G's and C's present, its length, the primer's concentration in the reaction and the sodium ion concentration in the PCR. A typical PCR primer is 20 to 30 bases long and ideally has a TM of around 55 to 65 degrees centigrade. So how do you design a primer with the correct melting temperature? Well, first you need to identify the region of DNA you wish to amplify and then look for primers sequences that are nearby and meet the criteria that I previously mentioned for a good primer. That is melting temperature, lack of hairpin loops and primer-dimer pairs, and specificity. Next, you calculate the TM of the primer, and there are two ways of doing this. One method, which is quick and dirty, back of the envelope type approach, and is rarely used, and the other, called the nearest neighbour, which is a more accurate method. In the quick and dirty approach, we count all the G's and C's in the primer and multiply by 4. And then count the A's and the T's and multiply by 2 and add the numbers together. This method gives a very crude calculation of the TM and, as I said, is rarely used. The other method is called the nearest neighbour method. And while you don't need to know how to do the calculation, you should at least be familiar with the theory. The nearest neighbour method uses the primer's length, the primer's composition, that is, what base is next to what base, hence nearest neighbour, the primer concentration in the reaction, and the concentration of sodium ions in the reaction. Once this is all fed into a program, the TM is calculated. Once you've identified a 20 to 30 base stretch of DNA that has the right TM, you then check to see if it can form hairpin loops and primer-dimer pairs. As the name suggests, a hairpin loop is a loop in the primer that forms as a result of the bases pairing to themselves. These loops can be tricky to melt and will disrupt the PCR and hence need to be avoided. Primer-dimer pairs occur when the primer can base pair with the other primer in the reaction. In this case, these primer-dimers may be amplified in preference to the template DNA, therefore leading to the reaction not being very efficient and hence a lack of product. The presence of hairpin loops and primer-dimer pairs are usually checked using a computer program. Finally, I usually do a quick blast with the primer to make sure it doesn't give a good match against another DNA sequence in the same species I'm working with. Or if the species is not human, in humans, as contamination of reactions with human DNA can be a problem. So that is it. That is how I would go about designing a primer in the lab. When working in the lab and doing PCR, you can sometimes have problems getting the correct material to amplify, and or you may need to add restriction sites to the product. So how can you do this? When you encounter low amounts of template DNA or are struggling to get the correct material to amplify, you can use a nested primer approach to improve yields and specificity. For nested primers, you use two sets of primers, with one set falling inside the other, hence nested. In the first round of PCR, the outer primers are used, and after this round of PCR, the reaction may not contain enough DNA or maybe a mixture of DNA molecules, some of which are correct and some that are not. The PCR products will then be purified to remove any of the leftover primers from the first round of PCR. In the second round of PCR, the inner primers are then used with the material generated from the first PCR. In the second round, there is more template available due to the first round of PCR, and the second set of primers will, hopefully, only amplify the correct DNA molecules, and hence improve specificity. Now, for adding restriction sites, the primers are designed with the restriction sites at the 5' end. However, the restriction sites presence means that there will not be a good match between the primer and the template, as the restriction site sequence will not match the template. To overcome this problem, the first few cycles, usually 5 or so, of the PCR are run at an annealing temperature around 10 degrees centigrade below the TM, so as to give the primers a better chance of sticking. The annealing temperature is then increased to the 3 to 5 degrees below the melting temperature, as there will now be template present that it can bind to. That is the template from the earlier cycles that contain the restriction site, and so the primers will match. Another trick that can be applied to help with the addition of restriction sites is a GC clamp on the 3' end of the primer. A GC clamp means that the last two bases of the primer are a G or a C, as these form a tighter interaction with the template due to each forming 3 hydrogen bonds compared to 2 formed by A's and T's. Hence the 3' end is more likely to stick. So there you go. That is how we add restriction sites to DNA and, using nested primers, overcome some issues around amplification of the correct product.